Madeira, Portugal. Um, this lecture is about basics of biomaterials and bionanotechnology, and I have mentioned it as small is wonderful as one of the subtitle because it is very interesting to see a large building, how it is built, how astonishing they are built and everything. But each large building is built by smaller bricks, and each smaller bricks have certain smaller atoms, right? So each atom are clubbed together to form a brick, so each bricks are aligned in certain way to form a big building. So it is more astonishing to see the smaller things, and if we understand the smaller things, it is easier for us to build a larger building. So that's why I put small is wonderful. So that's why today we are going to learn about smaller things, which is nanomaterials, and about basics of biomaterials, biomaterials, bionanomaterials, and bionanotechnology. Okay, fine. So this lecture series has three sections. So first session, I'm going to introduce what is nanomaterials, and in the second session, I'm going to mention about how to synthesize them, how to characterize them, and how to functionalize and formalize them, and how to utilize them in applications. And finally, I'm going to in the last session, I'm going to say about how to what is biomaterials, what is bio nanomaterials, and how they are helpful in bio nanotechnology. So without further delay, let's move towards the lecture. Okay, so the first session is introduction to nanomaterials. So what is a nanoparticle? A nanoparticle or nanomaterial, right? So if you reduce a particle, right? So if you have a particle with the size of about 1 to 100 nanometer, right? So it is a nanoparticle, right? So its size should be 1 to 100 nanometer, right? Okay, so what is nanometer? So 10 to the power of minus 9 of meter, right? So if you have a meter, so when you divide them like 10 to the power of minus 9, so those small particles are called as nanoparticles, and its size should be 1 to 100 nanometer, right? So if you have three dimensions, right, at least one dimension, right? So you have x-axis, y-axis, and z-axis. So if you have three dimensions, at least one dimension should be in 1 to 100 nanometer in size, right? So in some other cases, right, so in some other cases, especially in biological materials, bio nanomaterials or bio nanoparticles, if you have 1 to 1000 nanometer in sizes, right, so those particles are also categorized under nanomaterials or nanoparticles. So what is the actual definition of nanoparticle, right? So whatever be its size, 1 to 100 nanometer or 1 to 1000 nanometer, below microparticles, right? So below 1 micron, right? So whatever be its size, but its property should differ, right? So only if its property is different from of the microparticles, it is called as a nanoparticle, right? So its size should be 1 to 100 nanometer or 1 to 1000 nanometer in biological in bio nanomaterials, but its property should differ. Those are called as nanoparticles, and if you are building a material with those nanoparticles, those are called nanomaterials, right? So now what is nanomedicine, right? So we have conventional medicines, right? So like paracetamol or whatever, right? So those materials, those conventional medicines, when you when you convert them into a nano size, 1 to 100 nanometer in size, it is called as nanomedicine. So this is one case, right? So the second case is you take those conventional medicines like paracetamol and you formulate them inside a nanoparticle, right? You have a nanoparticle 1 to 100 nanometer or 1 to 500 or 600 or 1000, right, below micro, right? So its property is differing. So inside that nanomaterial, when you are keeping your conventional nanomedicine, when you are formulating them with a nanoparticle, that one is also called as nanomedicine, right? So these are the two definitions of nanomedicine. So what is the significance of this nanomedicine, right? So this nanomedicine right? So see, because its size is 1 to 100 nanometer in size, right? So this size is smaller, so because of that they are helpful in biomarker mapping, drug delivery, gene delivery, detection and diagnosis of several diseases, molecular imaging, targeted therapy, as I showed in this figure, right? Okay, why they are helpful in these biological applications? Because most of our biological macromolecules like mitochondria, ion channels, secretory granules, membranes, virus, DNA, and bacterial, the inside the bacteria ones, right? So everything is nanostructures, right? So for a nanoparticle it is easier to interact with this nanostructure, so that's why it is highly beneficial in biomedical applications, right? So now we are going to see about the history of nanomaterials, right? So in 1905 Einstein first said the sugar compound is about 1 nanometer, right? So at that time he cannot visualize them, so theoretically he said a sugar compound will be in the size of 1 nanometer. And he also mentioned that not Einstein, Newton also mentioned that when you reduce the size of a material, its property will differ. So for that he mentioned the example of gold. So gold, if it is reduced to smaller size, he doesn't mention nano size, but when you reduce the material into smaller size, solid gold will turn into liquid. So he mentioned that, right? So they have a theoretical idea of how nanomaterials will behave, right? But later in 1905 Ernst Ruska and Max Knoll, they constructed the first electron microscope. After that only we started to visualize nanomaterials, right? So we can able to see nanomaterial, but electron microscope at that time is quite premature, so people cannot be able to see what exactly a nanomaterial. A blurry image is appeared, but still they cannot actually see the particles. Right. So later in 1959, Richard Feynman, so he is known as father of nanotechnology. So he said that there is plenty of room at the bottom. So the title of the lecture is "There is plenty of room at the bottom." So this familiarized the theme of nanotechnology among researchers. Right, so this happened in 1959. Later in 1974, the term nanotechnology was given by Norio Taniguchi, a Japan scientist. Right, so he gave this term nanotechnology. After that only people started to call this technology as nanotechnology, and particles as nanoparticles, and nanomaterials, and everything started to come up. And later in 1981, scanning tunneling microscope was developed by Gerd Benning and Heinrich Rohrer, right? So they started to see more clearly how nanomaterials are forming, and how their structures are and how they behave. Later in 1986, Eric Texter wrote a book called Engines of Creation. So this familiarized nanotechnology among general public. Right. So now it is going from the researchers towards the public. Right. And finally, in 1991, Sumio Ijima invented carbon nanoparticles. Right. So these carbon nanoparticle invention is quite interesting, and it gained lots of companies interest, like industries started to be more interested in nanotechnology because of the arrival of C60 fullerene and everything, right? And in 2000, the National Nanotechnology Foundation, so initiative was created by in the US. So after this only, each and every country started to have a department especially for nanotechnology, and they started to give research fundings towards nanotechnology. Right. And 2004 Royal Society London, so they started their implication of nanotechnology in their medicinal field. And in 2009 only, nanoparticles are used to target for the targeted drug delivery systems. Right. So it started in 1905, but in 2009 only, people started to use nanotechnology uh, for biological application, especially for drug delivery. But even after this also, there is a bit of hesitation in providing approval for these type of drugs. So I will say why later, right? So in 2016, studies were moved towards from using the nanoparticles in biomedical application towards toxicity of nanoparticles, right? Okay, so now history of nanomedicines, right? So now we move towards medicinal one. So from 1964 to 1994, people started to conceptualize nanomedicine. So around that period, they were like telling liposomes or micro-sized particles, right? So liposomes, dendrimers, and polymeric materials, everything are microparticles. So they were conceptualizing like if you reduce the size, it will be more beneficial. They were saying this, and they were trying to utilize reduce their size, right? But from 1995 to 2007, they started to develop nanoparticles like liposome nanoparticles, nanoliposomes, nanodendrimers, and those things. And then they started to clinically validate and commercialize them, right? And now it is moving towards approvals, like they started to check their toxicity, and also they are sending them to approvals, and several particles are getting approved, right? So FDA is starting to approve. But even though if it started like 2005, the first approved FDA approved one was Doxil, right? So which is a liposome formulation of doxorubicin, which is an anti-cancer drug, right? So they started to approve, but still now we are checking more towards more nanomedicines, more nanoparticles are under the radar of FDA and EPA, European Pharmaceutical Agency. So European medical agencies and pharmaceutical agencies to get more to get approved even more, even for more applications, more biomedical applications, right? So these are the scenarios of nanomedicine. So until now we are moving towards the toxicity and approval regulations and those things, right? So okay, so now we are going to see about why nanomaterials are special, right? So as I said before, why in the first slide, so how I said because of its smaller size, they are helpful in biomedical applications, right? Because they can easily interact with nanostructures of our body or in the living organisms, right? So how they are interacting easily with nanostructures, right? So how nanostructures are easily interacting with nanostructures, right? So it is because of why nanomaterials are special. It is because of two things. One is its size, and second thing is its shape, right? So depending on its size, because of its smaller size, they have high surface to volume ratio. So what is high surface to volume ratio, right? So let's consider this one as a paper, right? So when you have this paper, and if you want to burn this paper, you are taking a lighter, and then you are burning this corner, right? So first this edge atom, consider this one as an atom, right? So consider this atom is interacting with the fire first, right? So for the fire to reach the center, it takes some time, right? So consider you are having fire on all the four sides, right? So only these four edge atoms will interact with the fire first, right? But consider cutting this paper into four, right? So when you cut this paper into four, instead of four edge atoms, you have 16 edge atoms, right? So instead of 4, you have 16 edge atoms. So all the 16 will interact with the fire. So instead of heating, instead of, let's say it takes 2 seconds to burn this, so it takes less than 1 second to burn the whole paper, right? So you are going to burn the paper anyway, right? But still, when you cut the paper into 4, and when you are doing it faster, right? So instead of taking 2 seconds, right? So the same temperature, same fire, right? So instead of taking 2 seconds, it takes less than 1 second. So this is what is happening in nanomaterials, right? So when you reduce the size, their surface is more, right? You have more surface with the given volume, right? So within the given volume, so that's why nanomaterials are having a enhanced properties, right? So that's why I said your bulk property or your microparticles will have property, so those properties have to differ when you are changing them into nanoparticles. Now, only then it is called as nanoparticle, right? So when you reduce them, their size, so its surface property differs, they have high surface to volume ratio, so that's why it is behaving differently, right? You have enhanced properties, so that's why they have their own properties or exclusive properties which will be beneficial for biomedical applications, right? So the best example for this is melting point of gold, right? So when you have the bulk gold which you are using them in jewelries and ornaments and everything, so the melting point of a gold, pure gold, is 1064 degrees Celsius, right? So when you reduce the size to nanometer, right? So for each ones like 50 nanometer and 40 nanometer, there is a huge difference, right? So the melting point differs from 1064 degrees Celsius to it can be reduced to even 200 degrees Celsius when you have 20 nanometer of gold nanoparticles. No, so its melting point is around 200 degrees Celsius only. So you can see the drastic change, how it is behaving, right? So this is because of high surface to volume ratio. So this property of nanomaterials, right? So the next one is one chemistry, many shapes. Right, so as I said before, two things are important in nanomaterials. One is its size, next one is shape, right? So just see this image, right? So you have several different types, several morphologies of one single material, zinc oxide nanoparticle, right? So you can fabricate them into rings, fibers, hexagon, like ribbon, like coils, like fibers, like whatever shapes, like nano flowers are there now. So you can see the crystals, right? So you can develop them into various types of materials, right? So various types of morphology, so each morphology behaves differently than the others, right? So it differs, it behaves differently with the size, and it also behaves differently with the shape. So that's why each nanomaterial is different, even if it is from the same material, depending upon its size and shape, its property will differ. That's why nanomaterials are very special, right? So now we are moving towards the second session, which is fabrication of nanomaterials, right? So for the fabrication of nanomaterials, two approaches can be used. One is top down approach, another one is bottom up approach, right? So top down is you have a bigger material, you are crushing them into smaller size, and then you are making a nanomaterial, right? So that is called as top down approach. Another one is bottom up approach. So in the bottom up approach, you arrange atoms, you form molecules, so those molecules form smaller crystals, so those crystals will form into a nanostructure, right? So one is from the top, you are reducing their size and then bringing them into nanostructure, and second one is they are smaller atoms, so you are arranging them into molecules, and then those molecules you are building them, right, from the bottom, right? So that is why it is called as bottom up approach. So this is a broad classification. So under this you have several types, right? So for the fabrication of nanomaterials, four procedures are followed. One is synthesis. You need to have a synthesis procedure, and then you need to characterize, you need to see their optical properties, their magnetic properties, their size, shape, surface properties, whatever, right? So everything you need to study, and then you need to formulate them. Right, so especially for biomedical applications, you need to formulate them to avoid its toxicity and to interact with other biological structures. And then finally, you have your finished product which will be useful for application. Right. So in the synthesis, right, so you have chemical approach, physical approach, and biological approach. Right. So basically, physical approach is top down approach, right? So physical approach, and some of the chemical approaches are top down approach, and many of your chemical methods and biological methods are top or bottom up, right? So this is for your general information. So why we have this chemical approach and physical approach, what are their benefits, right? So you have their benefits are, you can synthesize stable nanoparticles, right? So you can have stable nanoparticles, but you will use hazardous materials, which you cannot use them for biological application. So that's why we move towards biological approach. But the problem in biological approach is, even though their toxicity is lesser, the stability is not that much good compared to chemical and physical methods, right? So that's why now recently we have hybrid methods. So those hybrid methods are an intermix of chemical and biochemical biological approach, right? So we'll see that in detail in the next slide, right? So synthesis approaches, you have chemical approach, salt gel, co-precipitation, polyol, etc., right? So chemical approach, you have high stability, you can synthesize nanoparticle of whatever size you want, whatever shape you want, right? But their stability is also high, but they use a toxic reducing agent, right? So reducing agent is quite important. So in order to reduce their size, right? So in order to reduce their size, you need reducing agent. Those reducing agents, they use synthetic hazardous chemicals. So that's why we cannot use them for biological applications. For electronic applications also we can use them, but when you are in your when those electronic things, or when you dump them in the environment after their usage, they will also cause toxic reaction in the environment. So that's why chemical synthesized ones towards the toxicity is quite is not compatible. So next one is physical methods, right? So physical methods, you can have stable nanoparticles, you can have whatever size you want, but the cost of those materials like chemical vapor deposition, sputtering, scanning tunnel microscope, the cost is very high, so it cannot be used for large scale applications, right? So in lab scale it is okay, but for the large scale commercial applications it cannot be used. So that's why we move towards biological methods, right? So in this method we are not going to use high cost materials, high cost equipments, and we are not going to use synthetic hazardous chemicals, right? So we are going to extract biomolecules from bacteria, fungi, algae, or plant, right? So different parts of the plants you can extract, and you can also develop them, even like bacteria. Inside the bacteria nanoparticles will develop, and then we can extract the nanomaterials from the bacteria. So this is called intracellular and extracellular synthesis, right? So that's why we are moving towards this biological methods. But the problem in biological methods is stability, right? So its stability is very weak, right? So that's why currently we have mix of biological and chemical methods, which is salt gel synthesis with the help of biological extracts. So now we are moving towards that era, right? So what are sustainable nanomaterials? So the goal of bringing biological approach or biochemical approach, which is a hybrid approach, is to develop sustainable nanomaterials, right? So it should not cause toxic reaction in the living organism as well as to the environment, right? So that is sustainability, non-depletion of natural resource, and maintain ecological balance, right? So you cannot just go there and cut some plant and then utilize them for nanomaterial production. So it should be a sustainable development, right? So as I said before, physical and chemical approach uses toxic chemicals, and final product will also contain certain toxicity, right? So high and also the another approach is, so when you are going for physical and chemical methods, high energy, you need to use high energy like heat, pressure, right? So that's why we move towards biological approach, so which uses less toxic materials, so which is less toxic towards environment and other organisms, right? So however these materials are not stable, so that's why we move towards hybrid nanomaterials, right? And there is no regulation until now to measure sustainability of nanomaterials. right? So even FDA or European Medical Agency, Indian Nanomedicine Council, they don't have a perfect regulation for the sustainability of nanomaterials to calculate them. So now we need to focus on producing nanomaterials at low cost with low toxicity and without any toxic reactions towards humans and the environment for the sustainability in the future, right? And this sustainability is very important in biomedical applications, right? So until now, acute sustainability of nanomaterials can be analyzed, right? Even though there is no regulation, so this can be analyzed, but chronic sustainability cannot be analyzed, right? So we need to think about the future and how to calculate this chronic sustainability and etc in the future, right? So what are the ways to characterize nanomaterials, right? So if you have a material, right, so if you have a nanomaterial, it can be either in liquid form like colloidal nanoparticles or powder nanoparticle, right? So if it is, even if it is a, whatever be the type of your nanoparticle, either it is liquid or powder, right? So you can use all these methods like UV visible absorption spectroscopy to study its optical property, dynamic light scattering method to study its particle size distribution and zeta potential in a solvent, right? And then liquid X-ray diffractometer or normal X-ray diffractometer to know its crystallinity, and Fourier transform infrared spectroscopy to know its surface functional groups, and finally to know its scanning electron, to know its morphology. You can use either scanning electron microscope and transmission electron microscope, fine. But if it is a powder nanoparticle, to analyze its optical property, instead of UV visible absorbance spectroscopy, you will use diffuse reflectance spectroscopy, right? So this is the only difference, right? So from all these you can know what are the properties they have, like what are the initial basic properties they have, nanoparticles have, right? So for the formulation of nanomaterials, especially for biological materials, so if you have a nanomedicine or nanoparticle as a medicine, right, so you need to formulate them in order to deliver them at the target site, right? If not, other instead of targeted places, so it can be degraded by other areas. So for example, if you need your drug to be in the lungs and you are in your lungs, so it won't be going to lungs, so it will be everywhere in your body because it gets degraded faster, right? So in order to stop them, you need to formulate them. For the formulation, you can use either dendrimers, right? So dendrimers are here, they are hyperbranched polymers. So inside these void spaces, you can keep your medicine or nanoparticle, right? You can have micelles. So here you have micelles. So inside this micelles, you can have organic solvent. So you have a hydrophilic head and hydrophobic tail. So inside this, you can keep your nanomaterial. And liposomes, you have hydrophobic tails and hydrophobic bilayer and hydrophilic heads. So inside this, you can keep your hydrophilic core, hydrophilic nanoparticle, right? And you have and you also have core shell nanoparticle, right? So you have a core, right? So core nanoparticle, so over that you can also functionalize some biomolecules, so which is also a core shell nanoparticle. You can also use them, right? So these are all nano formulations, right? And finally, after all these procedures, you can move towards the final process before your application is preclinical and clinical testing. So for preclinical testings, you can use cell cultures and those biomolecular analysis, and then it ends in animal to human trials. So this is how you will develop your nanomaterials for any application, and especially for biological applications, right? So now we are moving towards the third session, which is bionanomaterials in bionanotechnology, right? So what is biomaterial? So now, until now, we know what is nanomaterials, right? So what is biomaterials? Say, so materials, those are extracted from biological organisms. For example, cellulose, chitosan, right? For example, cellulose. So this is a trunk of a tree. So from the trunk of the tree you can extract lignin, hemicellulose and cellulose, right? Right. So when you convert this biomaterial into a nanomaterial 1 to 100 nanometer in size, it is called as bionanomaterials, right? So just you are converting them. For example, nanocellulose and nanochitosan, as I mentioned here, right? So you can change them into a nanofiber, you can change them into a nanoparticle, whatever, right? So you can extract them, right? So the extracted ones are biomaterials, and when you are converting these biomaterials into nano-sized particle, it is called as bionanomaterials, right? So significance of bionanomaterials, right? So there are two types of bionanomaterials, right? So the first one is functionalizing synthetic nanomaterials. So you have synthetic nanomaterials, right? So from physical methods or chemical methods you have your nanomaterial, right? So in that nanomaterial you are functionalizing, you are attaching certain biomolecules on their surface, right? So that one is also called as bionanomaterial. And second one is converting biomaterials in nano size, like for example as I showed in cellulose and nanocellulose, right? So these two are also called as nanoparticles, bionanomaterials, right? So what are its significance? The first significance is less toxic, right? So they are less toxic, right? So they are less toxic, they are biodegradable, their bioavailability is higher, and they are easy to interact with microbes and cells, as I showed in my first slide, right? So easy to interact, right, because of its smaller size, and its bioreactivity is very high. So because of this reason bionanomaterials are widely used in biomedical applications, right? So currently there is a development in bio nano composites. Composites are blending of two materials, right? So for example you have one material A, so this has one weakness and material B has one weakness, right? So to have compatibility, right? So to have to enhance their weakness, to reduce their weakness and to enhance their positive points, you are mixing them together, right? So this is called as composite. So when you have a biological material and a normal material, and when you are blending them together, it becomes a bio nano composite, right? So the best example for functionalizing your synthetic nanoparticles is here, and for yeah, it's here. And for the best example for converting a biomaterial into a bionanomaterial is here, cellulose nanofibers, right? So in bionanotechnology, what are the benefits of this bionanomaterials, right? So you can see you can use them for n number of applications, right? But the first one which comes to every researcher's mind is drug delivery, right? So to deliver an anti-cancer drug, to deliver insulin, you are used it will be helpful for the treatment of cancer and diabetes, right? And the second one is implant, right? So either bone implants or dental implants, we can use bio nanomaterials because of its enhanced properties and application, right? And the third one is cardiovascular, to fabricate stents, right? So drug eluting stents are currently gaining more attention among researchers. So stents, for the stents also now cellulose nanofibers are highly are highly used, right? So next is neurodegenerative diseases, right? So the main problem in neurodegenerative diseases is when you are having a drug towards your brain, you have something called blood brain barrier, right? So there is a barrier which reduces, like which reduces the entry of certain drugs or certain biomolecules from the blood towards the brain, right? So those bionanomaterials has the property to bypass this blood brain barrier. So that's why for to provide drugs towards the brain for Alzheimer's disease, for Parkinson disease, so now people are using bionanomaterials. And biosensors, right? So biosensors to have even now for SARS-CoV-2, from the saliva to detect the presence of the virus, silver nanoparticles are used. Right, so it is not still commercially available, but still in the future, maybe next year, it will be coming out in market. So for the biosensors we can use, because it is highly environment friendly, it can degrade faster, right? And now people are developing antimicrobial fabrics. So these fabrics will kill the bacteria and viruses, since there are lots of variants of viruses, coronaviruses are there, and several other bacterial infections are also developing. So this antimicrobial fabrics will kill, so which will be beneficial for people who are working in hospitals, and in labs, etc. And for nutraceuticals, to for the controlled and targeted nutrient delivery for babies and mothers, and for the plants, for the animals, people are using bio nanomaterials. And for environment remediation, for wastewater treatment, for soil remediation, people can use bio nanomaterials, because it has high biodegradable application. And also finally, food applications, right? For food packaging, because it can degrade faster, instead of using plastic bags or plastic wrappers. So now people are developing bionanomaterials which can degrade faster, right? So that's all about my lecturer. So acknowledgements. So thanks to FCT, Fundación para Ciencia Tecnologia, Foundation for Science and Technology in Portugal, for funding my research. And thank you for your attention.